Hello friends, welcome again to my channel Asma's Botany. Okay, so today we will discuss about the new topic, that is the duplicate gene interaction. Okay, so duplicate gene interaction is a type of intergenic type gene interaction. So I have already discussed about the gene interaction type, that is the two types. First is the intragenic and second is the intergenic. So this duplicate gene interaction is the intergenic type gene interaction, means this is between the two different genes, and these two different genes can be present on the same chromosome on the different locus on the homolog 1 and the homolog 2, or on the non-homologous chromosomes also. So this is the third type. First I have discussed the modifying gene interactions and the complementary second, and the third type is the duplicate gene interaction. Okay, so what is the meaning of this duplicate gene interaction? We can understand it by the definition. The definition is gene 1, there are two genes, because we are studying the gene interactions. So for one phenotype, for one character, two genes are responsible. Okay, two genes interact to each other. Okay, so the definition is gene 1 and gene 2 have the duplicate relationships. Okay, duplicate means what? There are two genes. Okay, if the dominant allele from the first gene is absent, then only the dominant allele from the another gene is present. Then the dominant allele from any of these two genes can be able to and is sufficient to cause the dominant character. Okay, so this is known as the duplicate effect. Okay, so the two genes are duplicate of each other. Okay, so here you can see it is seen in the example, that is a Capsella bursa-pastoris. Okay, this plant is belong to the family Brassicaceae, the mustard family. Okay, and in this family, in this Capsella bursa-pastoris is also known as, the common name is the shepherd's purse, because its fruit is like the mostly triangular, flat, like purse like. So it is known as the shepherd's purse. Okay, so here the character is, what is taken is the fruit shape. Okay, so the fruit can be triangular in shape like this, mostly it is triangular, and top shape, two contrasting form of the characters. Okay, so to controlling this character, the fruit shape character, two genes are present. Okay, so gene 1 and gene 2. Okay, so the gene 1 have the two contrasting forms of the alleles. Capital A is dominant and small a is recessive, and here capital B is dominant and small b is recessive. This both the genes are controlling this one character. And the important thing, you must remember that this gene 1 and gene 2 have the duplicate effects. Okay, so first of all, if you do the cross in this, the parents you take must homozygous. So first this you take is a triangular shape of fruit parent crossed with this top shaped fruit parent. All the alleles are recessive in this, and in this all the alleles are dominant. So when form the gametes and in F1 generation, you can see the triangular fruits are produced because dominant alleles are present. But the duplicate gene interaction, actually what is the duplicate gene interaction, we will see only in the F2 generation to form this checkerboard. So for this we will do the selfing. Okay, in the selfing you form this gametes and then form these both form four gametes, and this also form four gametes. These are the, this, and then form the checkerboard. In this checkerboard you can see this is, so in this only one this genotype produce the top shaped fruit. But all have, this is the 16, and in the 16 only one have this top shaped fruit, but 15 genotypes produce the triangular fruit. Why? Because you can see, if the dominant allele is present, dominant allele means what? Only one dominant allele, and that can be from any of the genes, any of these two genes, or by the presence of only one dominant allele, and that dominant allele can be from any of the two genes. Okay, so this one dominant allele can form the dominant character. This is because this is duplicate to the another. Okay, so here you can see this is the triangular dominant character, this is triangular, and you can see this is triangular. But here you can see capital A is present, one gene dominant allele, first gene dominant allele is present, but the absence of the another gene dominant allele, but the phenotype is triangular because this have the capital have the duplicate effect. Okay, so here, and here also this capital B is present but capital A is absent, but here also the fruit is triangular because this capital B has the duplicate effect. So both the genes are duplicate of each other. Okay, only the condition when completely the dominant alleles are absent, then the production of the top shaped fruit occur only. This is only one condition. So in the F2 generation, the phenotypic ratio is the 15:1. 15 is the triangular type fruits, and mostly the fruits are triangular, and 1 is the top shape. Okay, so but the genotype remains same to the Mendel's dihybrid cross, but change in the phenotype. That's why we are studying it in a gene interaction. Okay, so I hope you understood. Stay tuned for the next videos, and if you like the videos, press the like button and share it, and don't forget to subscribe my channel as Smart Botany and press the bell icon for the latest update. Thank you.